that is a important thing for a species, is also being able to reproduce. So there's limited resources, and if certain phenotypes, we're talking about phenotypes, have a type of form that allows that individuals with that phenotype to more successfully utilize these resources, then they will increase in frequency from generation to generation because they are the ones who survive. And so that's a statistical outcome. And we'll look at some examples of how that goes on. Now, of course, what's really important is that any of this variation has to be genetically determined. If it is something that is acquired by exercise, then that's totally irrelevant because that's not written into the genes. And anything that you achieve by physical effort, exercise, the slate starts anew in the next generation. And I gave you several examples, modern examples of where natural selection has been working. Okay, then, of course, the modern synthesis, as we'll look at, is not only Darwin's ideas but of natural selection, but also Mendel's genetics. And this is the modern synthesis. Okay, now, I said that what I wanted to depart into next is some categories, some areas where we have evidence for evolution. And we're not going to have time to give multitudes of examples in each of these areas. I'm just going to give a few examples for these ideas. Okay, one thing about the evolution is that we can directly see it happening. Now, when I say this, we're not talking about observing a fish turn into a reptile, okay? That takes thousands and thousands of generations of incremental change. But nonetheless, we do see incremental change, and that can be explained by natural selection. And there's just a few of these I'm going to go into. What we can observe within our lifetime or within the generation of several generations is what we call microevolution, okay? And we can detect it both in the morphological observations of the changes in the phenotype of organisms, but also in their genetics. Now that, especially since the 1970s, where we have access more to genetic information. And so this change over short periods of time is referred to as microevolution. Opposed to this, or juxtaposed with this, is the term macroevolution. Macroevolution is something that we don't directly experience because we humans haven't been around long enough to do so. And that is in the realm of paleontology. So we need fossils to be able to reconstruct that type of evolutionary history. But still, microevolution is something we can observe. And so I'm giving one example here that's been well studied, something called the soapberry bug. Okay, these are bugs with a long proboscis. And originally they were a population where they first found in Florida, is way down in the south where it's a subtropical environment. So around Key Largo. And these bugs, what they feed on is a particular type of fruit. It was called the balloon vine fruit. And so these insects have a proboscis. Their mouth parts are a long tube. They have cutting edges at the very tip, and they will cut into the seed and suck out the oil that happens to be in the middle, okay? So this beak has to be a certain length. And of course, like a lot of biological phenomena, is that the length of the beak follows a bell-shaped curve, a normal distribution. There's some of these bugs that have short beaks, some of them have real long beaks, but most of them have a beak size somewhere in between. And if their beak gets to be too long, then they would drill right completely through the fruit and they would starve to death, okay? They're not thinking creatures like that. They just do it physically until the edge of the fruit comes up and touches their mouth. If their beak is too short, then it'll just drill into part of the fruit and not reach the oil. And therefore, bugs that have too short of a beak, they would die as well. So only those bugs that have a beak length that's within a certain parameters there can feed on these fruits. All right. Furthermore, this is not a single gene trait. Okay, so we're not talking about a single gene locus with 2 or more alleles per loci. It is the length is of the beak is determined by several genes. So it's polygenic. It's polygenic. Several genes come into play, each of those genes having 2 or more alleles. Well, sometime in the middle of the 1800s, due to human traffic, humans move around, and as they do so, they tend to move other animals and plants with them as well. And so a population of these soapberry bugs got established in the middle of Florida, okay, around this Lake Wales, okay? Now, their native food, namely this balloon vine fruit, does not grow there. Okay, it does not grow there. However, there is a related plant, and you don't need to know the names of these plants because like this one up here that's around Lake Wales, flat-potted golden rain tree fruit, okay? It's a long name there. It'd be hard to remember. Nonetheless, the point is that this is related to the balloon vine fruit, and therefore the type of oil that's there is compatible with the diet of these bugs, and so they could survive. However, one of the problems is that the fruit has a different shape, and therefore individuals that have real long beaks, which would survive just fine in Key Largo, their beaks were too long, and therefore they were eliminated because they could not feed on this fruit because their beaks would go all the way through. And therefore, this change from one host plant to another led to a selection for individuals that had shorter beaks. So the shorter the beak, then the greater the likelihood that they could feed on this fruit and survive. And therefore, after about 100 generations, when we look at, someone did a study and measured the length of beaks around Key Largo versus around Lake Wales, and the distribution of beak sizes has drastically shifted toward the shorter end. Yes, Alkazim.
So microevolution is only from the idea that these the species have short generations, and then we're able to observe it during our lifetime, or is
it? What microevolution is that we can observe changes within our lifetime. Okay, so we have to have an animal or a system where we can see several generations within our lifetime, or not just only our lifetime, but within the time span of scientific observations. Okay, and that's sort of hard to define. It depends on, it might be 2 generations of scientists or whatever. From the time we've recorded, we've looked at several generations of the same populations or related populations, and over time we see that there are collective changes. That is microevolution. Okay.
So, but we could only do that with something like a soapberry bug because they have short periods of lifespan, right?
Right. So we would have a hard time doing that with elephants, for example. Okay, because they have a very long lifespan, and so a generation of elephants or a generation of redwood trees would take a long time. So we observe this mostly. It's real easy to do it with bacteria, okay, because bacteria have very short turnover times, and other animals such as Drosophila flies, they've been used in these microevolution studies. But even bird species, especially over the last 30 years of observation, okay, that's within our lifetime that we can observe it due to climate change in England. Okay, certain species of birds are starting to arrive in England earlier in the year. And of course, because they're migratory, they go south for the winter, come back earlier. And it isn't just an adjustment intellectually, cognitively on the part of the bird, because the timing of arrival at their breeding grounds in England happens to be innate, okay? It's genetically determined, all right? But what happens here is as climate changes, the food that they feed on insects, okay? And so the insects are coming out earlier, and there's a big bloom of insects and before the birds ever get there, okay? In the past, before the climate started to warm, the timing of arriving in England, their breeding grounds, was fit with the peak of the insects, because this is what they fed on. But the insects are coming out earlier because their cues is based on temperature, not on sunlight, not on any innate programming in their brain. And so those birds that happen to arrive later, okay, normal, the insects are no longer there, and therefore they starve. But we still have a sort of a distribution of behaviors where some birds end up coming earlier, and then others come later, others come later. And so this has ended up favoring those birds that come earlier. And therefore they have been selected for because of the shift in climate, the shift in the times that the insects are there. So this is another example of microevolution. So yes, you're right. We can mostly observe microevolution in those organisms that we have shorter generation times, so that we can observe it. Otherwise, they outlive us. Does that make sense?
Yes, thank you.
Yeah. Okay, because here with the soapberry bug, we're talking about a change that's happened over about 100 generations of these bugs. So it wasn't instantaneous, but nonetheless, those bugs, so it's the environment, their resource that they were feeding on, it gave a selective advantage to those bugs that had shorter beaks. And therefore, this is the distribution of beak sizes, the bottom graph here, around Key Largo. Whereas this is the conditions that happens now around Lake Wales. So there's been a selection for shorter beaks. You don't have too many bugs that happen to be born now with too long of beaks. And so in order for this to have happened, there had to be a shift in the frequencies and combinations of the alleles that determine beak length. And so this is what microevolution is. And we have many, many examples that we could give for this. And I gave you a second one about some bird species changing their timing of migration. Okay, so that's one category. Another category from which we get a lot of evidence, and this, of course, this microevolution observations has been something that we've been observing within the last 100 years or so, okay, and been documented. So it's not just in the lifetime of one scientist, but in the lifetime of several generations of scientists from which we have records, okay, that are recorded. This type of microevolution has been going on for, ever, but it's only recently that we started observing and recording it. So Greek writers and Chinese writers, Islamic writers, they were making no sort of observations in this area, even though had they been doing so, we would have even a longer record by which we could observe microevolution. Okay, it's a new thing that we've been looking at. But even back in George Cuvier's time, okay, and Lamarck's time, however, what has been looked at is the commemorative anatomy of organisms, and that also gives evidence of evolution. We're looking at anatomical similarities in the body parts of organisms that are related to each other. They seem similar to each other, but these parts of the body have different functions. Perhaps one of the best examples here is the forelimb of land vertebrates, okay? This is sort of not a figure from your textbook, got it from somewhere else, but still, when we look at various types of vertebrates, frog, lizard, shame this is so small, bird, bat, and so forth, whale, human, cat, all these forelimbs are being used for different functions. Humans, We use our forelimbs. We don't walk on our hands. We use them to grasp and manipulate things, whereas cats and frogs, they use them for walking. Bird uses the forelimbs for flight. Just whale uses the forelimbs for swimming. Yet, despite that, all these forelimbs are located in the same position, and furthermore, they have the same underlying bone structure, regardless of that. Same underlying bone structure. From your shoulder down to your elbow, there is a single bone there called a humerus. And then from your elbow down to the wrist, there are two bones there. Then we have a collection of carpal bones, and then 5 digits, 5 fingers. And this is an underlying pattern that is true of all land vertebrates, and not just only land vertebrates, but a certain group of fishes that are called lobe fin fishes. In their fins, their pectoral fins in front, they also have this similar bone structure. So when we find these correspondences of parts, despite their shape, despite their function, these are referred to as homologous structures. Okay, so homologous structure, its definition is it's a similarity, anatomical similarity that is based on inheritance from a common ancestor. The reason we have this commonality of the structure is because the ancestor from which all these organisms have descended, they had that structure and we just inherited it. It changed a little bit, but still we have that overall pattern, and that is homology. This stands in contrast to, instead of a homologous character, something that's called an analogous character. In other words, analogy. Let me see if I can find my pen here. I have a new version. And it doesn't seem to, there we go. Analogous. Analogous characters are those that are similarities, anatomical similarity, that is due to convergent evolution. That means that they have a completely different evolutionary origin. And a lot of times their ultrastructure, when you look down, really analyze the structure, that they're built from totally different materials, a totally different design. For example, both insects have wings, and bats have wings, and birds have wings. Okay, so wings have evolved in insects, and in birds have evolved independently of each other. They function the same, they have the same function, and they have some convergent properties, because in order to fly, the wings have to have certain aerodynamic properties. And so there's been convergent evolution there, but what they're made out of is quite different, okay? And so that is analogous, convergent evolution. Yes, Avani?
It's called analogous characteristics?
Yes, analogous characteristics due to convergent evolution.
Okay, thank you.
Okay, all right. So, and furthermore, if we look across insects, from silverfish all the way to butterflies, they have legs too, okay? And of course, their legs are different in anatomical structure. They don't have any bones. And furthermore, the number of joints is quite different. The material out of which they're made is quite different. And, but within the insects, there's a lot of commonality. Okay, there's 5 different sections of their legs. And that is inheritance due to a common ancestor. Okay, Alkozim.
So homologous structures don't necessarily have to have the same function, but analogous structures do?
Usually, yes. Right, that's correct. Now, it isn't the case that homologous characters are always having different functions. For example, a pig walks on its forelimbs, uses its legs to walk, as well as a cat. And so their forelimbs have the same basic function, okay? And so homology, they could have the same function, but what is curious here is if they have different functions, what is the reason that they have a common underlying structure? Because legs could be made in any different way. Wings could be made in more than one different way. So why this commonality? It makes more sense if that commonality of the bone structure is due to inheritance from a common ancestor. It is explained by that. Okay, does that make sense?
Yes, thank you. But analogous always has to have the same function, right? Because you can't prove it's from the same ancestor.
Right. Okay. So give you an example here. I'm jumping a little bit ahead here, but it's still looking at the limb structure, 4 limbs of bats and birds. Okay. Is that homologous or is that analogous? It depends on what you're talking about. If you're looking at them in terms of organs for flight, function, that's analogous because the wings of bats evolved as a winged structure independently of the 4 limbs in birds. However, if you're talking about the bone structure of those limbs, that's homologous because both bats and birds have that humerus, 2 bones in the next joint, and 5 fingers. Now you say, I haven't seen any fingers in birds. Oh, but we'll get there, okay? Most of those fingers are there in the embryo, okay? In the early embryo, they're there. And one of those bones is lost, 2 of them fuses together. And I think, well, the thumb, there's still a little piece of it there in the wing structure of modern birds. But so it depends on what level you're talking about. As structures for flight, a bat wing, bird wing, that's convergent evolution, okay? It's analogous. Whereas the bone structure underlying those wings, that is homologous, okay? Does that make sense?
Yeah, thank you.
Okay. So another aspect of anatomy, oh, and incidentally, I'll go back here too to this, within vertebrates, the 4 limb, not only do we have the same underlying bone structure, but even when we investigate the muscle groups, even though some of these muscle groups from, say, like in a whale versus a bat versus a human, they have shifted. Some of those muscles have become very small because they're not being used, whereas others are large because within that particular forelimb, they have a certain function. But still, the different muscles even happen to be homologous. And why that similarity? It doesn't have to be a intelligent God, omniscient God could make a limb in any way they wanted. There is no reason why they all have to be the same. And so the best explanation here for the similarity is due to common descent. Okay, and let me try to get rid of that. There we go. Okay, another comparative anatomical feature to look at that makes sense in evolution, but not if these things were created by a god, is the presence of vestigial organs. Okay, what is a vestigial organ? It's an organ there that in related organisms has a function, but here it doesn't. Okay, for example, pelvic leg bones. Snakes don't have legs, right? How many snakes have you seen that are walking around on legs? But they're believed to have evolved from a lizard-like ancestor. Okay, so, but there are a few snakes that still have internal, they never project outside. Okay, so pelvic bones, your hind legs, your pelvic appendages. And so they have internally, they still have the bones there. They're very, very reduced in size and they're non-functional. Why put them there? Okay, you can also say that with the whale. It's whales, they evolved from land vertebrates, mammals, that eventually returned to the sea. And they have evolved into a more fish-like, for streamlining, that's very common. They still have their 4 limbs, and in their 4 limbs, of course, they have the typical bone structure of tetrapods, land vertebrates, but their hind legs have been lost, okay? But they're there. internally in the bones. They still have a pelvic girdle. They still have a femur, okay, which is the long bone in your legs, but it's very highly reduced and it doesn't do any good. And therefore, it is a vestigial organ. What can explain those? They make sense in terms of evolution because characters that happen to be non-essential anymore, they don't have to be used, then the underlying genetics structures, the genes that control for that, they can accumulate mutations over time and the structure becomes lost. Now, mutations that happen that change a structure that is essential for life and for survival impairs the organism, and what will happen is it may not survive to adulthood and may not reproduce, and therefore those mutations are eliminated. But if it is a structure that it doesn't matter whether it works or not anymore because it's not important, then it can accumulate over time mutations, and eventually we get vestigial organs. This is what will happen. I can give you another vestigial organ that's in humans, okay? It's a muscle that's at the base of your hair, okay? Hair follicles. So our skin has hair follicles, and this is true of all mammals. Now in hairy mammals, which most mammals have a lot of hair, what these muscles function as, it's a retractor muscle, but it will contract and pull the hair shaft upward, creating an airspace, you know, between the fur, the hair, and the skin to trap heat. Okay, so it's insulation. So when an animal is cold, it fluffs up its fur. It has a second function too, namely that when it's frightened, it's being attacked by some other animal. Puffing up the fur makes the animal look larger and therefore more threatening. And so those are the two functions of this muscle to elevate the hair follicles or elevate the hair shaft. We still have that muscle, and that muscle works just fine. But it has no function for us, because when it happens with us, it doesn't it doesn't keep us warm. Furthermore, it doesn't make us look bigger because we have lost a lot of our hair. All we get is goosebumps. That's all, goosebumps. Okay, that's what causes it, but it doesn't do us any good. Okay, so this too is a vestigial organ. Okay, and no, it's no longer functional. It works, but it's no longer serves any function. Okay, so this is another figure, once again, just looking at mammals. Once again, just mammals. Forelimb, okay, forelimb of a pig, forelimb of a dolphin, used to swim, and of a bat. And once again, emphasizing the same bone structure from shoulder to wrist to the humerus, two bones from the wrist to, or from, excuse me, the humerus is from the shoulder to the elbow, and from the elbow to the wrist, two bones, an ulna, a radius, and then we have these carpal bones and a pentadactyl. It's called a pentadactyl appendage, okay, meaning 5 fingers. Now you see here, we count only 4 fingers, digits in the pig, but in the embryo, the digit number 1 is there. It disappears. But and in whales, there's 5 fingers. In bats, 5 fingers as well. And we have 5 fingers as well. So this type of a pattern is quite widespread in mammals and also in other vertebrates, land vertebrates. Okay, so comparative anatomy, that's one area where we get some information, makes sense. But also embryology, the study of the changes from once we have fertilization of an egg, a zygote, it then undergoes certain changes. Now, with regard to animals, all animals, the earliest phases of embryology usually are very much the same, okay? Because we start out, first of all, as a zygote, right? Diploid, of course, zygote. And let me get a chalkboard here. In fact, I'll draw this on that once it comes up. Computer is slow. Okay, so we start out as a zygote, and this will then start to divide, and it becomes a ball of cells, about 16 cells. I won't draw them all in here, 16 to 32. It's a ball of cells that's called a morula. And this is only of animals, okay? It's not fungi or anything else. Then it will arrange, it keeps dividing, and then it becomes a large ball of cells that's one cell layer, it's a sphere that is one cell layer thick, and it's hollow on the inside, fluid-filled space on the inside. And this structure is called a blastula. And then what will happen is, as these cells continue to divide, is some of them will start moving inward, okay, in this ball of cells, creating an opening at one end and a second layer. And so that we have something that is two cell layers thick, still with a fluid-filled area on the inside. And this is called a gastrula. All animals go through this. From, let me get this over just a second. So I put gastrula there. Okay, and this is insects, earthworms, and other animals as well as the vertebrates. The early embryology is exactly the same. There's a little bit of nuances from group to group, but once we get beyond the gastrula, depending on the lineage of animals, then their embryology will depart. But if we look here at early vertebrates, paying attention to vertebrates, let me put up a figure here. It's from your textbook. It's a shame I could have put a fish embryo over here. So this is an early chick embryo beyond the gastrula stage. Okay, so this is much, much later. And when we compare, okay, I'll say this, Savannah, and then I'll let you ask your question. But if I had a fish embryo here, the early embryos of vertebrates all look the same, okay? Even the fish. So this here is a chick embryo, this is a human embryo. Now, what is similar here is that they all have a post-anal tail. In other words, the anus, what we mean by post-anal tail is it's a tail that, part of the body that extends posterior to the anus, okay? So this will eventually develop an anus up here. And of course, chickens don't have tails, nor do humans, but all cats and dogs do. Fish do too. But still, even though we don't have a tail, it is there in the embryo. Furthermore, there, in the pharynx region, that's the neck area, there are several pockets that are called pharyngeal pouches. Now in fish, and we all start out with the same number of pharyngeal pouches in this early embryonic stage. Now in the case of fish, those pharyngeal pouches will develop into gills, or at least the gill arches. Whereas in humans, chickens, since they're air breathers, they lose these pouches, or at least the pouches would develop in a different way. But still, they're the same early structures that you would find in the fish that turn into gills. And we still retain that. Why? Okay, and then after later stages of embryological development, then they can change quite a bit. Why do we have this? Because that was established in the early fishes. And to change some early stages of embryology, it will affect a lot of other later structures. So think about building a house. Okay, so they build a house. First of all, they have to build a foundation. Okay, foundation, and then they start building the framing and piece by piece. Now, if you make a change in the plans for the foundation, early on, then it's going to affect a lot of the other parts of the house. Okay, you have to change the plan for how many rooms there are going to be, the size of the rooms, where the electricity is going to be on the wall. Whereas if you keep the early parts of the plan, foundation, you build the walls, and you decide in the kitchen, once you have a kitchen there, that you really don't want the washing machine, dishwasher over in that corner and move it over to this corner, later changes are easier to do without affecting the whole building. But affecting some of the early parts that you put together in the building, if you change those, they will affect a lot of the other aspects of the construction later on. And that's sort of similar here with this early embryology. So with the vertebrates, it's the early stages of embryology that are very, very similar to each other. And that only makes sense in the light of evolution, that they've inherited this pattern from ancestors. Okay, Avani, I thought you had your hand up.
I had a question, but you answered it.
Okay, all right. So
if you don't mind, I had a question about the digital organs real quick. Is the appendix considered one?
early, it was believed to be that. The appendix it's a common structure in tetrapods. Do amphibians have it? I'm not real sure, but I don't know. It's part of something that's called a cecum. And the length of the cecum in various mammals really depends on their diet. For example, a rat has a very huge cecum. Something that is a mammal that happens to be an herbivore, such as a horse, have really big ceca. Humans, being omnivores and not eating a whole lot of fiber, the cecum is very, very small, and we mostly have this little piece that's called an appendix. And so it used to be considered that, but it still has a function. Okay, so since it has a function with the immune system, okay, because it's a place where white blood cells refurbish themselves, and therefore it really is not quite vestigial. It is reduced compared to even other primates. In humans, it is small compared to in a chimpanzee or gorilla, but nonetheless, it has a function with the immune system. So you really can't rightly call it a vestigial. So I wouldn't use that as a very good example. It used to be considered vestigial, but it isn't. Okay. All right. Now, this is more modern example area for evidence for evolution, namely in molecular biology. Molecular biology, the ability to really get down to the nitty-gritty of the genes themselves, the base sequence of genes, and even at the protein level, the amino acid sequence is something quite new, okay, since the 1980s or so. And it has been an area that has been really accelerating. And so this gives good evidence of evolution, because animals that are more closely, or any organisms, two species that are more closely related to each other, have fewer differences in the amino acid sequence of the proteins and have fewer differences in the base sequence of their DNA when we compare certain genes. And so this is a figure from your textbook compiling. It doesn't really tell me what DNA sequence this is, but oh yeah, I bet this is beta hemoglobin. So this is just one of the strands that codes for beta hemoglobin. In humans, chimpanzees, gorillas, mouse, ancestor. So the ancestor in a horse. So we see here that from mouse, gorilla, chimpanzee, and human, that at one position here, they all have a thymine, whereas the ancestor has a C, and a horse, which belongs to a different branch of mammals, has this C. And so this ancestor is probably something further down, could be a duck-billed platypus or something like that, something further down the evolutionary tree. And so at this position, the original was probably C. And what has happened in the lineage that branched off for mice, humans, and other primates is that there's a mutation, point mutation, where C changed to T, and therefore mouse, gorilla, chimp, and human have T at this position simply because that is where they've inherited from an ancestor. Okay, I'm trying to fix the sun here, right? Whereas within primates themselves, or in fact, deeper within primates, humans and chimpanzees, at a different position, the ancient form, ancestral form, nucleotide at that position was T, and in the ancestor of chimps and humans, it mutated from T to C. Humans and chimps have C at this position, whereas gorillas, orangutans, all other primates, they have T. And so then within humans themselves, still in the same gene, we have an A at a different position, whereas the ancestral position had C. And therefore, horse, mouse, gorilla, chimps, they all have C at this position, and it's only humans. Therefore, within the lineage leading to humans, there was a point mutation in which C mutated into A. And so right now, molecular evidence from DNA is one of the most powerful tools we have for determining evolutionary relationships. But this can also work. This can also work at the level of proteins. In fact, this was the first type of molecular evidence we had because it was easy to examine the amino acid sequence of certain proteins, homologous proteins, compared to DNA. DNA technology really took off a little bit later. And so this is a figure from a textbook looking once again, I believe this is, hemoglobin. This is probably beta hemoglobin. And if we're looking at the beta hemoglobin consists of 146 amino acids. So it's a relatively short protein, but we compare it from gorilla to rhesus monkeys to mouse to other organisms that are more distantly related to us and taking zero differences with humans. Obviously, what I talked about last semester is that in the hemoglobin, even within humans, we have a lot of different variants. But here, if we're taking a standard wild type sequence that is most common among humans, comparing it to the most common amino acid sequence in gorillas, we only differ at one major amino acid position. Whereas further away, monkeys, rhesus monkeys, we have 8 different amino acid substitutions compared to them. But further down with a frog, 67 different amino acid changes. And so the further we get away in the evolutionary tree, lack of similarity, the greater the molecular differences. And so this is good evidence of evolution as well. And it's one of the most powerful areas that we have for evidence of evolution. Okay. All right. Another place gives us evidence, and in fact, this is one of the major areas for Darwin in his time. Even though the fossil record was not very complete, it's still the fossil record, okay? In fact, Darwin, what he was an early evolutionist had to work with was primarily comparative anatomy and the fossil record. And even in his day, there wasn't all that many fossils anyway. And so, but since then, there has been a lot more fossils that have been discovered. So some of the evidence here of the fossil record, what do we learn from the fossil record? We learn the fact of extinction, okay? Things that God had produced, if they were created, extinction is hard to reconcile. Okay, why let something go extinct? But we have from the fossil record a lot of organisms that are no longer around. Dinosaurs and mammoths and saber-toothed tigers that disappeared from the earth more recently. And so the fact of extinction is very strong. And in fact, you go back further and further into the fossil record, the older material, is that we come up with organisms that are extinct and quite dissimilar from the type of life that we have running around on Earth right now. Furthermore, what we get is from the fossils, sedimentary rock. You remember that sedimentary rock are in layers, okay? And we have this observation, what is referred to as the law of succession, okay? And so the fossils in one particular geographical region, that the fossils are more similar to the plants and animals living in the region now than they are to plants and animals living somewhere else on the earth, okay, in different geographical regions. That makes sense. Consider Australia. If we're looking at the mammal fauna, what type of animals, collective group that I'm asking for, do you associate with Australia?
Marsupials?
Marsupials, okay? Exactly, marsupials, that's what we're looking at. So the fossils in Australia are also of marsupials, okay? Not of placental animals, all right? And this is what this law of succession happens to be. In South America, there are fossils of a critter that are called glyphodonts, okay? They're mammals. They were pretty widespread. Some of them were really big, some of them were really small, and they have anatomical similarities to another type of animal that is there in South America. There's more than one species of these called armadillos. Now, about 10 million years ago, when North and South America through Central America became connected together, this allowed a land bridge by which animals could move from North America to South America and vice versa. But that only was established about 10 million years ago. And so we do have armadillos in the southern part of the United, in Texas, for example. But still, the anatomy of the glyphodonts fossils, we don't have them around. They're fossils now. They're very similar to the armadillos. And this could be said of a bunch of other types of animals there. So the fossil record, in fact, from one layer to the next layer, to the next layer, to the next layer, we find a progressive change in the shape of the animals that is unique to that area. And this is what we mean by the law of succession, okay? Furthermore, now the fossil record has a lot of gaps, and simply because the Earth is very, very dynamic. And so certain sedentary rock that had been laid down in the past, because it was a sea basin or a big lake basin, may have become uplifted so that it's now dry land. And then it's subject to erosion. And therefore, some of those pages of the fossil record have been completely destroyed. And the Earth is dynamic, and so we're missing a lot of records. Some of the fossil record is buried so deep in the ground that we can't get to it, all right? And so we have a very incomplete fossil record. But also when we look at the major groups of animals that are existing today, we find gaps, okay? Because intermediates have gone extinct. And for example, fish versus tetrapods. We do believe that land vertebrates evolved from fish, okay? Furthermore, birds and dinosaurs, all right? We do believe that birds evolved from dinosaurs, a certain group of dinosaurs. But where are the transitional forms? Okay, those forms, they had to existed in the past. Well, the fossil record has them. Now, in Darwin's day, there was very few transitional forms, but he was certain that they would be found. And through many separate groups of organisms we have, okay? And I'm just going to give some famous examples. Okay, the connection between birds and dinosaurs. Birds are dinosaurs technically. So the dinosaurs really did not die out. Most of them did. Most of the branches obviously did, but there was one small group that survived and they evolved into birds. So there's this fossil bird that probably doesn't have any living descendants, but still it's an example of a transitional form in anatomy between a bird-like creature and a reptilian-like creature, dinosaurs. And this is called Archaeopteryx. And this is one example I do want you to know. It's very famous. It was about the size of a crow, okay? And okay, let's go here. So this here is the famous one from Germany. It was found in limestone in Germany, but very clearly, okay, it has feathers and long feathers, right? The impressions there. Furthermore, it sort of differs. Okay, let's look here at some of the differences here. In the mouth, it has teeth. Modern birds don't have teeth, okay? That is a dinosaur character. In fact, the teeth are very much like other types of theropod dinosaurs. Furthermore, it had claws on its wings. Modern birds don't have claws, okay? Except for there's one bird that does in the Amazon. It's called a what is the name of that bird? Quetzalcoatl or something like that. They're young, they do have claws on the forewings and they use them to climb trees. Those claws disappear when they become adults, but most other groups of birds, they never have claws on their forewings. They have claws indeed on their hind legs, but not on the forewings. But these birds did have it. Furthermore, another distinction here is that they had a long bony tail in which there's feathers on it. Modern birds don't have a bony tail. Okay, they have tail feathers, okay, long feathers that are sticking out, but they really don't have a long bony tail. So the long bony tail here is a dinosaur-like characteristic. So what connects them to birds is that they had feathers. And even that is not sufficient, because we have now, since Archaeopteryx, found several types of tetrapods, mostly from very good deposits. Hoatzin, that's the bird. Yes, that's it. Correct. I was in the Amazon 2 years ago, just before COVID came along, and I have pictures. I was in the Amazon of Peru, and there was a lake I was at, and I was able to, through binoculars, take pictures of a Hoatzin. And so I'll show, in fact, I probably have them in some of my lectures a little bit later on. I'll show them to you. That's correct, okay? So this is a transitional between a dinosaur and a bird, okay? It has characteristics. In fact, we have a close-up here of the wing, forewing. Once again, humerus, 2 bones in that area, and 1, 2, 3, 4, 5 digits with claws, okay? So this is a transitional. And furthermore, so that's birds, we believe, came from dinosaurs. Modern birds have lost the teeth. They have lost the long bony tail. There are some tetrapod dinosaurs, I started to say that, mostly from very good deposits, fossil deposits in China, where they have feather imprints, but they obviously could not fly. They obviously were not birds. And therefore, feathers is not a trait necessarily of birds. It already arose in dinosaurs. However, those dinosaurs could not fly. And so we do have these types of transitional forms. So I said that we believe that land vertebrates, so-called tetrapods, that'd be amphibians, reptiles, birds, mammals, that they originally evolved from a fish-like ancestor. And in fact, it's not just any group of fish-like ancestors, but types of fish that we have very few of them around today, but they were very, very abundant about 400 million years ago. And these are called lobe-finned fishes, okay? Do we have any intermediates between them? Yes, we have a pretty good series. It wasn't available in Darwin's time, but over the years, these fossils have been found. Yes, Aquazim.
I was wondering why people say chickens are related to the T-Rex specifically. Is that inaccurate or?
No. Okay, where did you get this?
I think it's like just like common knowledge people share like throughout grade school and stuff.
Okay.
People say that.
Yeah, well, really, it's T-Rex was belongs to a group of dinosaurs called theropods. Okay, and Velociraptor, that's a theropod. Microraptor, that's a theropod. So there's that's one major branch there. So we're talking about a big group of dinosaurs that we believe that birds came from. And so modern birds, they still have some anatomical features that relate them to what we can see in the fossils of theropods. Okay, so that's probably the connection there. Furthermore, there was a study. The reason I asked, how do you know this? Because there was a study several years ago, and it was highly, highly disputed, that the lab, I forgot her name, I think she was at the University of North Carolina or somewhere like that, but she and her team extracted from dinosaur bone. They were trying to get collagen. Okay, that's a connective tissue protein. And they claimed to have gotten protein from this dinosaur bone. It was a T-Rex. And when they determined the amino acid sequence of that collagen. Collagen is widespread, okay, in animals. However, the amino acid sequence of varieties of collagen does differ from one species to another. And so they compared that to the collagen amino acid sequence in a chicken. And they found that, well, not just only in the chicken, but to other animals as well. And in their study database, they were using the T-Rex collagen most closely aligned with that of a chicken. Okay, here we're not saying that chickens and tyrannosaurs are each other's closest relative. It means that it's putting together this big group of tetrapods being very similar to this big group of things that we call birds. It's just that it's based on a specimen of a tyrannosaur and a chicken, but we could use a songbird as well, and it would work just as well as a chicken. Does that make sense?
Yes, thank you.
Okay, so I'm rather surprised the connection between a tyrannosaur and chicken specifically, when it's really that theropods and birds. Okay, birds are considered a subgroup of the theropods, but I would be very surprised that anyone would draw together a Tyrannosaurus and a chicken if they didn't know anything about that collagen study. But anyhow, it's out there. Yes, okay. Now, going from fish to tetrapods. This is Darwin's day, no intermediates. We have a lot of them now, okay? Tiktaalik, this was discovered in Alaska, or no, it was in Yukon. Probably about 10, 12 years ago, I want to say. Tiktaalik belonged to a group of fish that we only have very few representatives alive today. They're called lobe-fin fishes. This stands in contrast to most common fish that you are aware of, salmon, pike, and catfish. Those are not lobe-fin fishes. They're called ray-fin fishes, and we'll talk about the distinctions between those a little bit later on. But nonetheless, Tiktaalik has a lot of similarities, and it's between an amphibian and a fish. And so looking at Tiktaalik, this is an artist rendition of what they probably look like. This is the basic fossil. Now, modern fishes, fishes have continued to evolve, and therefore the comparison of the bone structures in the head, we can only compare it to Paleozoic fishes. Okay, fishes that lived, 300 million years ago. And so a lot of the anatomy of this Tiktaalik is very much like Paleozoic fishes, lobe-fin fishes. Lobe-fin fishes have the same bone structure in the forelimbs and in the hind limbs, the fins, as modern tetrapods. Okay, they have one single bone up here, corresponds to a humerus, connected to two bones below, and usually with a lot of finger-like supports in the distal part of the fin. And here on the Paleozoic group of these intermediate creatures, is that they have not just only 5 fingers. You find all combinations, anywhere from 3-digit like bones to 12, but somehow in the transition of these critters to land tetrapods, it was reduced to 5. Okay, why 5? We don't know. Could have been totally accident. But nonetheless, this here has some characteristics that I'm not going to go into that make it more similar to Paleozoic amphibians that were fully on land and Paleozoic fishes, okay, in between. So it's a good intermediate. And furthermore, there's, I'm not going to show other slides here, but there's later versions, later than Tiktaalik, that still have a lot of fish-like characters. Okay, other transitional forms. Okay, Aquazim, is your hand up or did you just leave it up?
My bad.
Okay, yeah, okay. Now, I do want you to know Tiktaalik, and I do want you to know Archaeopteryx. With Tiktaalik, I'm not going to say too much. I didn't say too much. I just said it's very good in its bone structure, intermediate between a amphibian and a fish, without pointing out. I did point out the bone structures in the limbs. Okay, that's common to both tetrapods, land vertebrates, and lobe-finned fishes, and that's homologous. But I gave several characteristics here with Archaeopteryx that I do want you to know. How is it more like modern birds? Not because it has feathers, but because it has flight feathers. I said there were dinosaurs that had feathers, no question about that. The function of the feathers in dinosaurs, we believe, probably could have been mate display, okay? Because we have some fossils where they just have some few feathers just only on the limbs, and they obviously could not fly. The feathers were not in the same form as flight feathers in modern birds. But examining these wing feathers in Archaeopteryx, they're definitely flight feathers. And so it isn't the possession of feathers that makes it a bird, it is the possession of flight feathers, okay? That is how it's similar to modern birds. Okay, so question up here, do ostrich have flight feathers? Yeah, they have flight feathers, but they've lost the ability to fly. They're too heavy for one thing, and the flight feathers are somewhat reduced, but they still have the same structure, because what really makes it a flight feather is not that it is used for flight, but because of a certain form. They're rather asymmetric in the way the veins are coming off of the main shaft. And so there's a bunch of anatomical things about the feather that makes it a flight feather, okay? And it's very obvious with the muscles, pectoral muscles and everything in the ostrich, that they came from an ancestor that did have the capability to fly. And there's other flightless birds too, like a penguin, okay? And in fact, there, does it really have flight feathers? Well, the feathers are so modified, I wouldn't necessarily call, they still have a similar structure, micro ultrastructure as flight feathers, but they use their forelimbs to swim completely. So evolution still makes these experiments and goes off of the main lineages. And so, okay, another idea about what the feathers were for is that probably a lot of tetrapods, we don't know about like stegosaurs and others that are further down the evolutionary tree or on a different branch, but of the tetrapods that had feathers, another possible function besides mate display might be thermal regulation, okay? Because this is another group, just like mammals, we maintain a constant body temperature. Birds do too, okay? And therefore, these may be thermal regulatory structures. So someone is here is what are some ancestors of peacocks? They're still basal in the birds, but I'm not real sure of exactly what group of what bird group the peacocks belong to, and that's probably, we don't want to go into too much details, but I'm not even sure what they're related to, but still, they still came from birds that fly, and in fact, peacocks can fly, okay? And I'd have to look up exactly where peacocks belong in the group of birds, and I'll do that, I'll find out, okay? So going back to Archaeopteryx, yes, I want you to know what is common with modern birds that ties it to modern birds is flight feathers. What makes it similar to ancestral dinosaurs is it has claws on the forewings. Not that it has claws, because all birds have claws on their hind legs. It's in their forewings. That's what's significant. They have a long bony tail. Modern birds do not. And they also have teeth, which modern birds do not. Okay, so those are intermediates. Okay, so whales. Whales look very much like fish, all right? But we know that they are mammals. They produce milk, they have hair, believe it or not, although most of that hair has been lost. Furthermore, a lot of the skeleton features tie it very much to land mammals. So how did they come from the land and make the transition back into the ocean? In Pakistan, a very well series of fossils, all well dated, you know, and I don't expect you to know these. All I'm wanting to say here is that there's a good fossil record. Whales have certain aspects of their bone anatomy that is only found in whales, and yet some of these early forms that were fully terrestrial with long legs have that bone feature. And Diacodexus, one of the older ones, and then Ambulocetus, something much, much later. And then when we get down here, the Durodon, it is fully aquatic and so forth, and it's lost a lot of its hind legs. So there's a good fossil series that ties whales to a land ancestor. And so we do have these transitional forms. Okay, all right. So yes, there's a lot in fossils that still support. Darwin didn't have these available in his day. And so these are, as we accumulate more and more fossils, we're finding this type of evidence. Biogeography also provides evidence of evolution versus creation. What is biogeography? It's the study of the distribution of organisms. Why are they where they're located? Why do we have certain types of plants and animals in South America that are not found in Australia? Or why are there certain groups of plants and animals on the Hawaiian archipelago that you don't find anywhere else? And so it's an attempt to try to understand the distribution of these groups of animals. And so the study of biogeography also sort of makes sense with evolution, okay, biological evolution. Okay, so if we look at two different islands that, let's say they're tropical islands. Tropical islands, they have the same basic climate. They have the same annual temperature, the same annual rainfall, precipitation, but they're located in different parts of the world. One of them is in the tropics of South America, and another one is in Southeast Asia, and they're about the same latitude, etc., having the same environmental conditions. But when we compare the plants and animals on those two islands, we find that they are not the same, not even similar to each other, except for by convergence, convergent evolution maybe, but not by what we see as homology. And in fact, the island in tropical island in the Caribbean, and close to South America, its closest living relatives are found on the mainland of South America, whereas in the island in Southeast Asia, it's those plants and animals have their closest relatives, closest counterparts on mainland Asia. Okay, that makes sense in terms of evolution with descent, with modification in the same location. All right, so furthermore, when we look at larger land masses, yes, Alpizim.
So just to clarify, the conversion evolution is just like specific traits that are being optimized for that environment that they'll share?
Yeah. For example, we've seen this time and time again with ocean animals, is that they develop sort of paddle-like limbs, okay, that look like fins. Their bodies become very smooth and streamlined, okay? This is because swimming in water is not like walking through air, okay? It has a lot of resistance, and therefore to cut down on the energy expenditure of being able to swim in ocean waters, then there's been this convergence to a certain body shape, okay, for streamlining, and also for limbs that can further push water so that you can move. And so there's been, all fish have that type of body, but also whales and dolphins, they came from tetrapods, mammals, land animals, going into this environment. They have converged on that same body shape. And that's because the environment selects for it. Although the streamlined body of a fish, say it's like a salmon, versus that of a whale, it's the shape and the fin structure or leg structure, paddle-like, that is convergent. But yet the underlying structure where that came from is different. They have different ancestry.
Thank you.
Yeah, okay. And so I'll give another example of convergence. This is in the New World, in the Americas. In deserts, plants have a tendency to reduce their leaf shape into spines, cacti, for example. And furthermore, the stems become very, very thick and can store water. Okay, this is favorable for living in a desert where there's a lot of, where it's very dry, hot, reducing the size and, or reducing the surface area of the leaves will lead to less water loss. And so cacti, the leaves are the spines, okay? And they don't really function all that strongly in photosynthesis. It's the stem. Stem being nice and thick, it will store water. Okay, those are adaptations for being in a very dry environment. But there's also deserts in Africa, in the Kalahari Desert in East Africa, or West side of Africa, that there are plants there that are not related to cacti at all, but they have developed a similar type of morphology because it makes sense with the environment, okay? Because they've reduced the leaf size to spine-like structures to reduce water loss. Their stems are very succulent, and so forth. And so that's convergent. But when we look at exactly the structure of those leaves, those spines, the structure of other parts of the plant, we can tell that it's convergent, and it's due to being subject to a similar type of environment. Okay. All right. Does that make sense?
Yes, thank you.
Okay. Now, furthermore, if we're looking at larger land masses, we'll just compare Africa versus South America. Okay, there's many different types of habitats, biomes, like a desert biome, tropical forest, savanna, okay? And when we compare the animals in a desert in South America with the animals in a desert in Africa, they're not each other's relatives. And in fact, the closest relative to a South American desert animal you would find somewhere else in South America, maybe in the mountains in the Andes, or it might be in the tropical rainforests in the Amazon, or its closest relatives that are similar to it by anatomy might be in the Pampas in the south, in Argentina. So even though we have the same types of biomes, desert, tropical forest, savanna, grasslands, the animals on both of these two, and even the plants, this is even true of the plants, in South America and Africa, their closest relatives are in other biomes on the same continent. That makes sense in terms of evolution, okay? Because ancestors lived there and they diverged in the same region, even if they specialized to become desert animals or plants or tropical forests. They're still in the same basic region. And so this study of biogeography also makes more sense with evolution. Now, some of the things before I leave this, so these are broad areas where we have a lot of evidence that makes sense with evolution, but not with special creation. I want to be careful here, though. One thing, if we have an omnipotent God, he, she, it, or they can create the world any way they want. And so you can't look at the world and just say, rule it out, because it's very possible that such a creature, such a divine being, could have made the world exactly as we see it, all right? It can't be ruled out. However, that's untestable as far as supernatural powers, and it's outside of the realm of science. Science is looking at what we can understand with what natural laws that we can see. So still, this all makes a lot of sense with biological evolution. Now, this idea of biological evolution has been rejected by a lot of people by claiming, oh, it's just a theory. It's the theory of evolution, okay? I want to dismiss this idea of theory versus law, okay? In science, when we have something that's called a theory versus a law, it tells you more about the time in which it was proposed. And so we have Newton's law of motion, but Einstein's theory of relativity. Yet Newton or Einstein's theory of relativity explains better the motion of objects than Newton's law does. And in fact, it supersedes it. So this idea in science of theory versus law really tells you nothing, okay? Earlier scientists tended to call their ideas laws, whereas later people referred to them as theories. Furthermore, the common use of theory is not the same thing as a theory in science. Words have different connotations. And when we talk about theories within science, a theory is a body of knowledge, connected facts that explain a lot of material. And various theories may have more support than others. So just denigrating the idea, saying and rejecting it is based on the theory, then you're doing a linguistic fallacy because you're not using the word theory in the way in which scientists mean it. Okay, so, okay, beat that horse to death. So we go on. Is it non-testable? It is very testable. Microevolution can be tested, and many labs have done that with bacteria and subjecting them to certain conditions and watching them evolve. Furthermore, even with paleontology, macroevolution, is it testable? Yeah, but not in the same way that you can do tests within a laboratory with some other types of laboratory sciences, because there you can set up and do manipulative experiments. But it was predicted in Darwin's time, even, and a little bit after, that there should have been a creature that was intermediate between a fish and a tetrapod. Okay, now you can't just go out there and look for it, but it's still, that's a prediction. That's a prediction that the theory makes. And it isn't that you can set up a manipulative experiment, but nonetheless, it makes a prediction that there should be fossils there. They may have been destroyed, but by finding a fossil that is intermediate between a fish and a land vertebrate, that is a test of the theory, and it provides evidence for it. And so there's various ways in which the science works, and this doesn't render evolution non-testable. It is very quite testable. Okay. All right. Those are the areas in which we have evidence for evolution. Now let's just look more deeply at this modern theory of Darwin and Mendel put together. So evolution takes place in populations. Okay, individuals don't evolve. The smallest unit that can evolve is a population. And populations, what are populations? They are local interbreeding groups of organisms of the same species, all right? But individuals do not evolve. And of course, this involves interactions between the environment and genes, okay, that determine structure. Okay, let me see here in chat. All right, yeah, just Wi-Fi. It looks like everyone's okay. So Aquazim, you leaving your hand up or do you have a question? Yeah, that's what I thought. That's all right. I just want to make sure when I see a hand up that I answer. Okay, so so I'm going to concentrate here, first of all, on microevolution. What really is changing in here? Okay, well, the evolution per se. When we're looking at macroevolution, which is we can only detect with fossils. However, we're starting to move into the area where we can even test hypotheses on macroevolution from the DNA of present animals and plants. We can do that. It's heading that way. But nonetheless, still, most of the evidence for macroevolution comes from fossils, paleontology. And so what's changing here is morphological characters over time. Looking at the transition of vertebrates from water to land, that we're looking at limbs, fins, and lobe-finned fishes that transformed into legs, and along the lineage to dinosaurs from legs into wings. Okay, so those are the types of morphological change, and it's not overnight. It takes many generations for that to happen. That is the area of macroevolution. With microevolution, and of course, even with macroevolution, DNA is changing piecemeal as mutations arise, and those mutations are then tested by the environment, all right? Selected for if they're beneficial, selected against if they're detrimental. And we'll look more details at that a little bit later on. So with microevolution, as I said, we can see evolutionary changes in the here and now, okay, within our lifetime, within those organisms that have short turnover times, and this is referred to as microevolution. I gave the example of the soapberry bug, but there we were concentrating on just the anatomical features of the length of the beak. But we can also look at the changes in their genes over time. So for microevolution, it is a branch and a part of biology. It's called population genetics. Population genetics is studying the change in genetic structure of a population. And so this is where we're going to start looking at population genetics, microevolution. Some definitions here before we get into microevolution is population, because microevolution takes place in populations. Populations is a localized group of individuals, interbreeding individuals most of the time, of the same species, but it isn't the species. The species is made up of many different populations, okay? So we might talk about a population of California poppies that's on a certain location on Mission Peak. Okay, so you go up on Mission Peak and you find a big cluster of poppies, and then there's grass everywhere else. And then way over there, there's another group of poppies. Well, that one cluster of poppies constitutes a population, okay? So of the same species. So the genetic structure, it's impossible to look at the entire genetic structure of a population. A lot of times when we're looking at what we call genetic structures, we're examining the genetic structure based on just only a few gene loci. And that's what we're going to do here, concentrating on the genetic structure of a single gene locus. So it's a genetic makeup in the population. So how do we describe the genetic structure? I'm going to give you 3 different ways we can look at it. Let's say that we go out and we have a species of plant, a flowering plant, and so there's 2 different phenotypes, red flower, pink flower. Actually here, 3 phenotypes, red flower, pink flower, white flower. So some individuals produce red flowers, some produce pink flowers, some produce white flowers. So first of all, before I go too deep into here, I want to make sure that everyone understands what we're talking about when we say genotype, phenotype. What do we mean by genotype?
How it's expressed in the genes.
We could get a little bit more precise than that.
Like alleles?
The allelic makeup at a gene locus, okay, whether it's homozygous, heterozygous, homozygous dominant, heterozygous, or homozygous recessive, okay? That is the genotype. Now, how does that relate to phenotype? What is the phenotype?
How it's physically expressed.
The physical expression of those allele combinations. So here we're talking about phenotypes, red flower, pink flower, white flower. Now I want to emphasize I'm talking about one gene locus, okay? Because some characters, including flower color, it can be polygenic. Okay, that the character is determined by more than one gene, but here we're just sticking with just one gene locus. And let's say we go out into a population of this particular species, And we don't pull up everything. We just need to have a big sample size, and we count randomly. And we find that we have 32 that are red, 96 that happen to be pink, and 72 that happen to be white. Furthermore, this particular gene locus has only 2 alleles, big R prime. And so it is a case of incomplete dominance, so that big R is not dominant to R prime. They're both equally expressed. So we have 3 different genotypes that correspond to 3 different phenotypes. So we have red, and their genotype is big R, pink, where the genotype is big R prime, white, big R prime. So our sample size here is 200. Now, based on this data, what we can do is we can calculate what's called the phenotypic frequency of our population. We're not looking, unlike when we underwent and studied last semester Mendelian genetics, we were making predictions as to the ratios from 2 breeding individuals vertically. Here, we're looking at the structure of the whole population. We don't know who is the parent of whom and who are brothers and sisters or whatever. We're just looking at the structure of the whole population, some of individuals which might be more closely related by descent than others. Okay, so phenotypic frequency, all this is one way to describe the genetic structure. It's not the best way, however. Simply, it is what proportion of this population is red, what proportion of this population is pink, what proportion of this population is white. And so we can do this very easily from our data. So the phenotypic frequency of red occurring in this population is simply the number of red individuals divided by the sample size. So 32 divided by 200, 0.16, we want to turn that into a percentage, and therefore the phenotypic frequency here is 16%. For pink, it's the number of pink individuals divided by the sample size. So this phenotypic frequency of pink individuals is 0.48 or 48%. 48% of the flowers individuals happen to pink, and likewise with white, the number of white individuals divided by the sample size. And notice here that all of these frequencies have to add up to unity. Okay, so if we keep this as a decimal form, 0.16, 0.48, 0.36 is equal to 1. Okay, it has to add up to unity. So this is the phenotypic frequency, and it gives us some idea, but it's not the best description. Another way in which we could describe this population's genetic structure is by something that we call genotypic frequency. Okay. Genotypic frequency is the percent of individuals in the population that have a particular genotype. So we're not looking at the phenotype. Well, we determine the genotype by looking at the phenotype, but still we're concerned with the genotype. So in other words, which percent of the population is big R, big R? Well, those are the red individuals, all right? So we have 32 red individuals, the big R, divided by our population size, and we come up with the same frequency as phenotypic frequency, all right? 0.16. And genotypic frequency for big R prime, 0.48, and big R prime, 0.36. So when we have incomplete dominance, the phenotypic frequencies and the genotypic frequencies are the same, all right? And I'm starting here because we're going to get more complicated as we go on, all right? Now, and once again, these add up to 1, okay, when they're in the decimal form. Even this genotypic frequency is a little bit more useful, but there's something else here that is even more better, is what is called the allele frequency. Okay, now with the allele frequency, we're asking which percent of all the alleles in our sample happen to be big R, regardless of how they're combined, whether it's in big R or big R prime. What percent of all the total alleles of this gene locus in our sample represent big R, and also big R prime. So we're looking at just only two numbers here for allele frequency. So here we can use some mathematical symbols. Let's let P represent the allele frequency for the big R allele, and Q the allele frequency for big R prime. And therefore we have to look at our, calculate what P is. So first of all, let's look at the red ones. How many of the alleles in the group of red, how many big R alleles do we have?
32.
We have 32 individuals, but how many big R's?
64.
64. Okay, and because there's each individual has two. And in the pink individuals, how many big R's do we have?
96.
96. In the white individuals, how many big R's do we have?
0.
144.
0. Okay, so P is going to be equal to 2 times the number, okay, 32, plus the 96, divided by twice the sample size. Okay, and that turns out to be 0.4. Now, I want to point out that we can do something else here. This is also equal to, we could also do it here mathematically, is that it's equal to 32, because we have 32 individuals that have 2 big R's, plus 1/2 of the pink individuals, 96, divided by the sample size, 200. We can do either way, and it gives us the same number. It gives us the same number, okay? Because here we're looking at 32 individuals that they only have big R, and then of the pink individuals, there's 96 individuals, but they only half of their genotype happens to be big R, and then our sample size. And these give exactly the same thing. Okay, you can do it either way. And so Q, how many big R primes do we have among our red ones?
0.
0. And the pink ones?
96.
96. And the white ones?
72 times 2.
Yeah, 72 times 2. And
144.
therefore divided by twice the sample size, and it gives us 0.6. Note, so these are the allele frequencies, and this is even more useful, as we will see. So allele frequencies, genotypic frequencies, these are the best ways to describe the genetic structure of the population. And so notice here that P plus Q equals 1. Okay, this is where I'm going to stop, and then tomorrow we'll go further and with this allele frequencies. Okay, any questions? Any slides you need to see? Okay, so what we'll go into tomorrow is that, okay, 1 o'clock tomorrow. What I'll do, of course, I'm not going to lecture for 3.5 hours. Okay, so I'll continue the lecture for about, what would be a normal time for a lecture. Then I'm going to talk about Monday's lab a little bit, because I'm wanting to put you into groups of 2 and have one of you go to campus. I'm trying to arrange it for Friday to pick up a little kit, a genetics kit that we'll be using, okay? In each group, I'm going to divide you into 11 different groups, and therefore 11 of you, once you have your partner, you're going to have to decide which one of you will go to the campus to get the genetics kit. And that will be all for tomorrow. Okay, any questions, anything you need to see?
Yeah, can I see slide 21?
Slide 21.
Okay, thank you.
Okay. Anyone else?
Could I see 20, please?
20.
Okay, thank you.
Okay. Anyone
Can I
else?
see slide 7?
Slide 7. slide 7.
Okay, thanks.
Okay, anyone else?
I had a quick question about the groups. Are you dividing them yourself, or do we give preference?
You can choose.
Okay.
Okay, and in fact, we'll have to do that tomorrow because right now you can't. The email is unreliable. Okay, and so I would take a poll. We'll do that after lecture tomorrow. Okay, but if you have preferences, think about it, because I'll put together whomever you want to work with, and those of you who do not have any preference, I will randomly put you together with someone. And we'll work that out tomorrow.
Okay. Have a good day, Professor.
Okay, all right. Okay, if there's nothing else, then have a good rest of the day. Nice and sunny. Get out and get a little of the sun.
Professor, I have a question.
Sure.
For the tecalic, the lobefish, and then the tetrapod, what is the similarity? It's like the limb structures?
Well, the skull structure and the limb structure, yes, very much limb structure. But one of the things here is that the limb structures are more advanced than what you have with typical lobefin fish. And probably Tiktaalik lived in water quite a bit, but in very shallow water, so that it had fins. It could swim probably, but it could also walk.
Mhm.
Because it's due to, of course, you can't really see the muscles that are operating the fins, but still a lot of times muscles are leaving little, what I call them, lesions, little spaces there that you can tell that that's where the muscles were attached to. And so you can deduce from that a little bit of how they were using their limbs. And so they were being able to walk in water, and they probably were even able to walk on land.
Mhm.
But they still, from the fossil, it still looked like they had fins. They weren't fully developed legs, but they were getting very close to that.
Okay, thank you.
There's other fossils I'll show a little bit later. I forgot what its name, Eustathanon or something like that. This one here is actually an amphibian. It's definitely on land, but its leg structure and skull structure is very, very similar to Tiktaalik.
Mhm.
So it's not just Tiktaalik alone, it's also, a series of fossils that we have from that time period.
Okay, thank you.
Right? Okay, anything else? Okay, so have a